Hi, everyone. I've got 1 o'clock Eastern Standard Time. This is the last session of the virtual conference, although there are other activities and things going on. And so this is population genetics and genomics. And I just have to make this big enough so I can read the whole thing, and connectivity. So our first speaker that's up is Rachel Finch on the population structure of Black skimmers in North America. So take it away, Rachel.
All right. Hi, everyone. My name is Rachel Finch. I am a senior majoring in biology at Eastern Connecticut State University. And today I'm here to share my research on the population structure of Black skimmers in North America, which I performed under Dr. Patricia Stish. To get us started, I just want to highlight why we're all here today. Whether researchers, educators, or students, we all share a common interest and a unifying goal, and that is conservation. So conservation, as you all might know, is the protection and management of biodiversity, as well as the efforts implemented to prevent the future loss of biodiversity. While genetics focuses on the mechanisms of heredity and the interactions of genes in the environment. When we combine these two, we are able to look at the dynamics of genes in populations, as well as the changes in the genetic diversity and structure of populations over time due to environmental, biological, and social factors. These then influence the efforts we take in order to help protect these fragile species. So when we think of protecting fragile species, the IUCN red list might come to mind, especially with endangered and threatened species. However, as you can see in the figure on the right, the black skimmer was considered a species of least concern as of 2016 by the Red List. This essentially means they're grouping together the birds in North America and South America together. However, in North America, the American Bird Conservancy lists the birds that reside in New Jersey as endangered and those both in North Carolina and Florida as a species of special concern. While none of these evaluations, whether at the state or global level, place a big emphasis on genetic factors, and thus they focus solely on demographic and geographic data. And because of this, over the last few years, a few papers have emphasized the need for augmented genetic indicators. And so why is genetic diversity so important? Well, although genetics starts at a microscopic scale, genes have a macroscopic effect and thus should be included when evaluating the status of a species. Genetic principles are useful in assessing conservation priority as well as risk assessment, and especially when it's difficult to evaluate using only demographic or ecological data. Effective population size, for example, is crucial in both evolutionary and conservation biology because it influences the rate of inbreeding as well as the loss of genetic diversity, the efficiency of natural selection, and the maintenance of evolutionary potential. Population structure, or the distribution of genetic diversity among populations, is highly variable and is shaped by a variety of evolutionary and biological processes. Many of these processes, such as gene flow, for example, you can see in this diagram below, is greatly dependent upon dispersal and is influenced by environmental and social conditions. So as we look at this population, as we look at the species of black skimmers, there's a lot known about the natural history of them. However, only one other study has been performed on the genetics of black skimmers. And this was done in South America on the South American subspecies of black skimmers, and it was done by Mariano and his team. And this paper was actually published in the Waterbirds Journal. So this study has provided a baseline for genetic diversity and differentiation at a continent scale. And we are happy to acknowledge that now with this study in our own, we're able to report genetic patterns across the whole hemisphere. And then we hope to differentiate populations genetically. So for the North American black skimmer, we know that their breeding range encompasses North America and Central America, from Southern California down to Northern Ecuador on the Pacific coast, and then from Massachusetts down to Northern Venezuela on the Atlantic coast. We know that the populations in California and New York follow a north to south migration route, while those in Texas follow an east to west migration route. We also know that the birds in the Atlantic in Texas are highly migratory, while those in California have a minimal migration route. And those in Florida are actually residential. And we know this thanks to mark and recapture data. And although we know a lot about the breeding grounds of this species, less is known about their wintering grounds, and thus leads us to think that the non-breeding grounds and staging sites are areas for intermixing and gene flow. So when we began our study, we had a few goals in mind. The first being that we wanted to understand population connectivity using molecular techniques. So we wanted to assess genetically how related and different these populations were to each other. We then wanted to apply what is known about dispersal patterns, and we used mark and recapture data, and then we hope to fill the unknowns of this data using our genetic data. With these goals in mind, we formulated two hypotheses. The first being that there would be a greater degree of population structure in individuals breeding in different oceanic basins. So we believe that the California population and the New York population would have a greater degree of population structure, while Texas would have more of an intermediate population structure. Our second hypothesis was that population structure will be strongly correlated with migratory strategy. In this, we see that New York and Texas, we assumed would share more alleles, being that they both had a very highly migratory nature as well as potential for intermixing during the non-breeding season, while California would be less inclined to share the same alleles or as many of the same alleles due to their relative geographic isolation as well as their minimal migration. So when we began the study, we had a total of 73 individuals sampled by Kate Goodenough and her team, and these samples came from San Diego Bay, California, Corpus Christi, Texas, and Nickerson Beach, Long Island, New York. And you can see the exact numbers of samples we had from each site right here. We then implemented 3 different types of DNA extraction, as we had whole blood in liquid buffer. We had feather samples, and then we had dried blood on PTA filter paper. We initially used 8 individuals, which we genotyped at 25 microsatellites. And of these 25 microsatellites, only 9 of the loci amplified successfully and were polymorphic, which we then used to genotype all 73 individuals. And it's important to mention that of these 9 microsatellites that we used, 5 of them were used in the South American study. And then after all of our genetic data was collected, we were able to calculate genetic indices and population differentiation. So here, looking at table 1, we quantified 3 different measures of genetic diversity across the 9 loci that we used in our study. And here you can see we use the mean number of alleles in each population, the observed heterozygosity, as well as the expected heterozygosity, and then the fixation index, or the inbreeding index, as it's also called. And here we see that the California population has the least amount of alleles present compared to the other 2, as well as the least amount of heterozygosity observed. And thus, they have the smallest or closest to 0 value for the fixation index. This demonstrates that the California population has the least genetic diversity among the 3. However, since they all have negative fixation index values, this shows that there is gene flow and outbreeding occurring. We then look to population differentiation, and we did this using FST values. And just a brief overview to familiarize you all, FST values are a pairwise comparison, and therefore the values are relative to the populations, and they range from 0 to 1, and the closer to 0, it means the more similar the populations are. So as we look at the boxes outlined in green, we see that New York and Texas have very small differentiation values compared to California and New York, or California and Texas, outlined in red, which show greater differentiation. And although that these values are low compared to other populations, we still see a greater difference between California and New York and California and Texas. And this correlates back to our second hypothesis, being that New York and Texas would be more likely to share more alleles due to their migratory behavior. We then wanted another way to evaluate population differentiation, and we did this using the program structure. And this allowed us to estimate the best number of gene pools or genetic clusters given our data pool. And here you see in the first figure above, when we first ran the structure program, it clustered as 2 or 3 gene pools. And as you can tell, it's pretty hard to differentiate which one fits our data the best. So we ran a subsampling analysis, and this subsampling analysis allowed us to even our sample size to 16, which, if you recall, Texas and New York had almost double the amount of samples than California did. So as you see in the figure below, the subsampling analysis allowed us to smooth these clusters and basically supported a cluster of 2 compared to 3. And this also tells us that the pattern above is likely due to the uneven sampling size. And then we can draw this back to our FST values we saw in the previous slide, showing that New York and Texas had very little differentiation, while California had a more greater distinct population differentiation when compared to New York and Texas. So as our genetic results aligned with mark and recapture data, we were able to make 2 new observations. And the first being that we know the California population was first confirmed breeding in 1976. We are seeing a population that is highly isolated compared to New York and Texas, as well as it has a limited dispersal range. As I mentioned, it had a smaller migratory distance, as well as minimal gene flow. And this could likely be due to the founder effect. And then to bring back this map here, our samples from Texas actually came from 2 distinct sites. And we know that when, thanks to mark and recapture data, we know that when the Texas birds leave their breeding sites for the winter season, they don't go to Florida. They actually stay in the Gulf Coast, not past Louisiana, or they go to the Pacific side of Mexico, thus demonstrating a split migration strategy. However, the birds that do go to Mexico have the potential for mixing with California birds. Our data does not support that theory. It actually further supports the isolation of California birds. So being that our study relates back to the South American study, we wanted to compare our genetic diversity back to the 5 loci that were analyzed in the South American species. And here we see that whether we're dealing with our subsample or our full sample, we have a larger sample size that we were dealing with. However, the number of alleles present in both cases were smaller than those in the South American subspecies of black skimmers. And we also had less, smaller values for heterozygosity, both observed and expected, which also is another indicator of less genetic diversity in the North American skimmers. We then moved on to compare the population differentiation among the 5 loci, and we see a similar result. As you can tell here, there's very little differentiation among the North American skimmers, while down here, the South American skimmers, the values below this diagonal have not been corrected for null alleles, and thus they actually had significant values. And although they're not significant after the correction of null alleles, they're still more clear and distinct differentiation. And the reason we see this is likely due to the fact that the North American skimmers are highly mixing and have more movement in North America compared to South America. And so as we continue our research, we hope to increase our sample size as we're working with a group of widely distributed but genetically connected populations. And we hope to increase our genetic database as we do this. We also want to increase more selective sampling. As I mentioned, the birds in Texas had the 2 distinct breeding sites and that split migration strategy. So we'd like to tag birds that we know go either to the Gulf Coast or to the Pacific side of Mexico, and then overlay the tagging data with genetic data to see if there's differentiation or variation among the Texas population itself. And then, as I had mentioned, the Florida population is residential, so we'd like to see if the Florida birds are highly differentiated compared to our other populations in North America. And we're also receiving samples from Peru once they can get here. And Peru is considered the Amazonian subspecies of skimmers, which we'd like to compare to the North American and the South American subspecies eventually. And we'd like to monitor the effects of environmental changes such as the development of beach property, which beach nesting sites are really important for black skimmers, and as they become more residential, and as well as climate change progressing and rising sea levels, and see how those affect the gene pool we're working with. And we're currently working on a manuscript for our study, which we hope to submit to Waterbirds by the spring of 2021. And I'd just like to take a moment to thank everyone, the Waterbird Society, for hosting this online conference, despite everything going on with the pandemic. I'd like to thank Dr. Patricia Stish, as she's been my mentor for the last 2 years at Eastern. And I've been a part of 2 different Waterbird projects now because of her. I'd like to thank Kate Goodenough, as she collected all the samples we used for our study. She's from the University of Oklahoma, and she is also playing a major role in the manuscript, as well as helping with natural history of the birds. David Newstead from Coastal Bend Bays and Estuaries program. He's also helped a lot with the mark and recapture data, as well as the natural history, and he's participating in the manuscript as well. And then Brittany Valicani, who spent countless hours collecting this data with me, and then Eastern, Connecticut State University, for giving me this opportunity to work with Dr. Stish.
Thank you, Rachel. Excellent talk. So let's see, there's a question in the chat. Do you have any plans to sample the Atlantic coast skimmers from New Jersey to Florida? And if you are, or would you expect to find a greater genetic diversity from the birds from New Jersey to Florida?
I don't think we looked into sampling New Jersey down to Florida. I know we thought about Florida itself because they're the residential population. For genetic diversity, I don't know how different they would be from the New York birds, but we thought the population structure itself would be different.
Okay, and then Dave Moore asks, which North American population is most closely related to the South American birds?
Well, the South American birds, when they ran the structure program, they had 2 clusters as well as we did, and they were divided more as a Brazil population kind of, or a Brazil and Argentina population. And I'd have to go back and look at the study published by Mariano to give you a more accurate answer for that.
And then there's a question on what kind of buffer you used to collect and preserve your samples, and how long the samples were preserved before running the test on them. We did not collect the samples, so I'm not exactly sure, but I could ask Kate for that. Okay, that's a question for your co-author. And okay, so any other questions? I guess I have a question on what is constituting a designation as a subspecies between North and South America? Is it strictly a geographical isolation?
I think
answer this if you want me to a little bit. It's all morphological. There's not been any other genetic comparisons, so it's all basically just morphological. The South American species have different coloration on their wing pattern, with the wing patterns and tail pattern, and they're bigger morphologically in bill and tarsus length than the North American species.
What does the genetic data support having them be a different species?
Well, as of now, we know they're a lot less diverse, so potentially they don't share the same alleles.
Okay, thank you so much for your talk, Rachel. And next up, I'm just going to get to my notes here. is Delini Aberama, and I hope I'm not butchering your name there, Delini. So if you can share your screen.
Yeah, you got my name. Thank you.
That's great. And you are ready to go.
Yeah. So thank you so much. Good afternoon, everyone. I'm Dilini Aberama, and I'm here to present our research on two birds of a feather flock together, population genetics of yellow-nosed albatrosses. So seabirds are important. Why do we think that seabirds are important? Because seabirds are apex predators in the marine environment, and also they are key indicators of the marine health around the globe. But unfortunately, during the past few decades, the status of the seabirds has deteriorated, and most of the species has come close to extinction. And also, most of the causes that cause population declines of seabirds are due to human activities. So what could be these human activities that cause our seabirds to close to population decline? So among among the all threats, the main threat is the longline fishing, which happens all around the world, and this longline fishing affect almost all the types of seabirds, and also as we can see in the figure, there are different kinds of fishing happens throughout around the world, and ultimately most of the seabirds follow these vessels to catch food easily, and they get caught in the hooks and get drawn in the sea and end up dying. And also another threat is the introduced species such as house rats, and also infectious diseases such as avian cholera. So most of the seabirds get affected by all these threats, and these are few main threats that they face. And due to all these threats that are very difficult to avoid, we think that the conservation of seabirds are crucial. So as the study species for our study, we selected the two yellow-nosed albatross species, the Atlantic yellow-nosed albatross and the Indian yellow-nosed albatross. These two species are a good fit for our study because both of the species have a limited limited number of breeding sites, and both species are having a decline in their global population size. And IUCN has identified both Atlantic and Indian yellow-nosed albatrosses as endangered, and specifically there's a disagreement in the taxonomy of the two species because some of the taxonomies identify these two species, two groups as two different species, while some other taxonomies identify them as two subspecies of the same species. So, and also when you look at these two birds, we can say that they look much alike and look similar, but there are some differences between these two species. So when we talk about the morphology of the two species, in the Atlantic yellow-nosed albatross, the color of the head is more gray colored, while it is more white in color in the Indian yellow-nosed albatross. And also the eye patch of the Atlantic yellow-nosed albatross is more dark and is triangular. But in the Indian yellow-nosed albatross, the eye patch is not that prominent and it's not that sharp. These two features, the color of the head and also the shape of the eye patch, can be used to distinguish only the adult birds of the both species. But the other two features, the shape of the culminicon or the yellow colored strip on the bill, and the naricon, the horny segment that covers the nostrils of the bird, can be used to identify or distinguish the juveniles of the both species. So the difference is that in the Atlantic yellow-nosed albatross, the base of the culminicon is more rounded at the base, while in the Indian yellow-nosed albatross, the base of the culminicon is more pointed at the base. And also, when looking at the naricon, or the horny segment that covers the nostrils, in the Atlantic yellow-nosed albatross, this horny segment is more curved, while it is more straight in the Indian yellow-nosed albatross. So using these two features, culminicon and the naricon, we can differentiate even the juveniles of the two species. And not only the morphology, but there are differences in their breeding range. So as their names suggest, the Atlantic yellow-nosed albatross breed on a few islands in the Atlantic Ocean, and the Indian yellow-nosed albatrosses breed on a few islands in the Indian Ocean. And the numbers in the within brackets represent the number of mature individuals on each of these islands. And so not only the breeding sites, but also there's a difference between the difference in the foraging range of the two species. So as you can see in this map, the red dots represent the foraging range of Atlantic yellow-nosed albatross, and they are more confined to the Atlantic Ocean, while the foraging range of Indian yellow-nosed albatross, represented by the green dots, are more confined to the Indian Ocean, and there's a minimum a small overlap between the foraging range of the two species around South Africa. So we obtained this data from the BirdLife International seabird tracking database. And interestingly, not only the two species show differences in their migration, but some populations of the same species also show differences in their migration. As an example, the Atlantic yellow-nosed albatross breeding on the Nightingale Island fly mainly to east, and the Indian Atlantic yellow-nosed albatrosses breeding on the Guelph Island migrate either east or west. And so these are the two islands that we have data for in the BirdLife International tracking database. But according to data from the researchers who monitor these birds on these islands, they say that the Atlantic yellow-nosed albatrosses breeding on the inaccessible island fly only in west. So this is an interesting case for the Atlantic yellow-nosed albatrosses. And in these maps, the blue color stars represent the approximate location of the breeding islands. And based on these facts, we developed three main research questions for our study. So are the Atlantic and Indian yellow-nosed albatrosses genetically distinct? And do the differences in at-sea distribution correspond to the genetic differences between and within species? And can we use bycatch samples to determine the at-sea distribution of the two species? In order to answer these questions, we analyzed 354 samples from four breeding sites, and two bycatch locations. So for the Atlantic yellow-nosed albatross, we had 23 individuals from Inaccessible Island, 42 individuals from Nightingale Island, and 136 individuals from the Guelph Island. And for the Indian yellow-nosed albatross, we had 33 individuals from the Amsterdam Island. And as for bycatch samples, we had 38 bycatch samples from seas around South Africa, and we had bycatch samples, 82 bycatch samples around New Zealand. So, and when we genotype the bycatch samples using species-specific primers, we identified that all these bycatch samples were Indian yellow-nosed albatrosses, So we included them as two different populations of or two different groups of Indian yellow-nosed albatrosses in our further analysis. So for all these 354 birds, we genotype them, or we analyze them using both mitochondrial and nuclear markers. First of all, we analyze them using the mitochondrial control region. So based on the haplotype network we constructed using the mitochondrial control region sequences, we identified two clusters representing the Atlantic yellow-nosed albatrosses and Indian yellow-nosed albatrosses. One interesting thing happened here, and there was one Atlantic yellow-nosed albatross that clustered with the Indian yellow-nosed albatrosses, and to see whether there was an error during sequencing, we genotyped the same individual twice, but both times we end up with the same result. And when we look at the genetics or genotypes of this individual for the nuclear markers, we found that this individual has the genotypes that is correspond to the Atlantic species. And using mitochondrial control region, excuse me, we couldn't see any differentiation within the species. And then we analyzed our samples for single nucleotide polymorphic sites within the OCA9 nuclear region. So in that, it showed genetic differentiation for the two species. So most of the Indian yellow-nosed albatrosses were homozygous for A, while most of the Atlantic yellow-nosed albatrosses were either homozygous for G or heterozygous at the particular SNP. And interestingly, in this nuclear marker, we were able to see some genetic differentiation within Atlantic yellow-nosed albatross. As you can see in this bar plot, most of the individuals from Nightingale and Inaccessible Islands were heterozygous for, with a G and an A at the particular SNP, while most of the individuals from the Guelph Island were homozygous for a G. So with the idea of analyzing, looking for more genetic differentiation between and within the two species, we analyzed our samples with 13 microsatellite loci, and we ran our data in structure software. So with the structure analysis, we found two genetic, so all these structure plots are shown at k equals 2. And with the structure plot also, we found two distinct genetic clusters for Indian and Atlantic yellow-nosed albatrosses when we analyzed all our populations together. There we wanted to look into population differentiation within species. Therefore, we analyzed the structure plots or microsatellite data separately for the two species. So when we looked at only the Atlantic yellow-nosed albatrosses, we found two genetic clusters clustering individuals from Nightingale and Inaccessible Islands together and separating the individuals from Guelph Island. So when we look at the geographical location of these three islands, we could correlate this genetic differentiation into their geographic location, because the two islands, Inaccessible and Nightingale, are about 20 kilometers apart, while the Guelph Island, which is about 400 kilometers apart from the Nightingale Island, which is the closest island to the Guelph Island. So here we could see the differentiation in Atlantic yellow-nosed albatross. And when we look at the Indian yellow-nosed albatrosses, and we could see that all these three groups were genetically different. When we analyze all three populations together, the biket samples from the were different from both Amsterdam Island as well as the South African bycatch samples. And same for the South African bycatch samples, they were different from Amsterdam Island population as well as bycatch from the New Zealand. And therefore, we came into conclusion that these two bycatch groups, South Africa and New Zealand, were not originated from the breeding island Amsterdam. And at the very first, we could separate bycatch from New Zealand as they were significantly different from Amsterdam and South African bycatch groups. But when we look at the first structure plot for between South African and Amsterdam Island groups, we were not sure whether there's a significant difference or not. Therefore, we took only the individuals from Amsterdam and South Africa, and we ran the structure analysis just for these two groups. And there we were able to look two distinct genetic clusters for Amsterdam Island and South African bycatch samples. So, so when we look at the number of breeding individuals and on each of the islands for breeding of Indian yellow-nosed albatrosses, we can see that there are only 3 islands that harbor a considerable number of breeding individuals: the Prince Edward Island, Crozet Island, and the Amsterdam Islands. So since bycatch samples from South Africa and New Zealand are different from Amsterdam Island, we removed that island, and then we wanted to look whether we can trace the origin of these bycatch samples. Therefore, out of the 4 of the remaining breeding sites, we can see that the Kerguelen Island and the St. Paul Island would not be able to survive a loss of 38 or 82 individuals. Therefore, we removed these 2 islands also. And then we had Prince Edward and Crozet Islands. And according to a study from McHaddo in 2018, they found that birds breeding on Prince Edward Island mostly forage in around South Africa. So considering this information, we conclude that the bycatch samples that caught around South Africa originated mainly from the Prince Edward Island, and our other group, the New Zealand bycatch samples, were originated mainly from the Crozet Island. So, so from our study, we saw that the 2 species, the Atlantic and Indian yellow-nosed albatrosses, are genetically different, and also the populations of both species are undergoing differentiation. And since both these species are identified as endangered species, we say that the genetically distinct groups within these species also should be treated as separate conservation and management units. And as we could track the origin of the bycatch samples with our study, we proposed to use these molecular studies to and the bycatch samples to track or monitor the birds from islands that are not monitored regularly throughout the time. And at the end, I would like to thank all these people who provided us samples and also provided funding for our research, and especially for the members of the Berg Lab of University of Lethbridge. And time for questions. Thank you.
Thanks so much, Delaney. Super interesting study. So there's about a minute for questions. I guess I would say hopefully we could just get rid of bycatch sampling. It's like get rid of bycatch altogether, so we're not getting
yeah.
albatross.
Unfortunately, it's like really difficult to get rid of it because the fishing is happening all around the world, legally, illegally, both. So
right. Okay, there's a question. Has there been any records of hybridization in nature for these 2 species?
So for these 2 species, we haven't seen any records for hybridization, but so the breeding locations are highly distinct for the two species, but there are about 2 cases where they have seen a pair of Indian yellow-nosed albatrosses on the Guaf Island, but they haven't shown any breeding there and no records of hybridization.
Okay, thank you. I would just say keep your eye on the chat, Delaney, if
Yes,
anybody
sure.
else puts questions in there, and because I've got 140 here, so I'm gonna queue up if we can queue up the next person.
Yeah.
So that's
you.
Paige, so thank you so much.
Hi, everybody. Sorry, I'm trying to figure this out. So, not sure. I've done this so many times. I'll just share my screen.
There you go. It's coming.
Okay. Very good.
You have to double click to enter full screen mode is what it says, Paige. We don't quite see your slides yet, so you just say double click to, there you go. Now we see it. I see him now.
So we're good?
Yeah, we're good.
All right. Thank you. Thank you everyone for coming, and thanks so much to Waterbird Society for hosting this online meeting. I'm very excited to be here today and presenting some results from chapter 3 of my dissertation, looking at connectivity of roseate terns, not just in North America, but actually across the Atlantic Basin. So we've expanded our scope a little bit here. So Rachel and Delaney have really set me up well for this talk, talking about genetic diversity. And so here today we're talking about population connectivity. And population connectivity in this context is really referring to the permanent one-way movement of an individual from its natal site to a new population where it then breeds. So that breeding aspect is very important. Connectivity among populations is very important for the persistence of wildlife populations and the persistence of species. And that's because connectivity among populations is important for promoting gene flow between those populations. And gene flow is necessary for promoting adequate levels of genetic diversity within populations. And that genetic diversity is very important for promoting the evolutionary potential of populations, so their ability to adapt, to respond to changing conditions with habitat or with climate change, etc. And when this connectivity among populations is severed, populations can become isolated, they can lose the evolutionary potential. Smaller populations can become subject to inbreeding depression, and all of this can contribute to population declines. So maintenance of connectivity is very important. So here on the right, we have this kind of classic metapopulation matrix where we have 4 breeding populations of varying sizes and with uneven or asymmetrical rates of gene flow among them. But then humans come along to ruin the party, as we often do. So humans are very good at disrupting these historical connectivity regimes among populations. So now we've built, let's say, a road between populations 2, 3, and 4. Whatever the species is, say a black-capped vireo does not disperse over roads. So now we've severed population 4 from that connectivity regime, and we've isolated it. And say genetic diversity within this population erodes over time, and eventually we lose this population. So this is a very real process that we see within metapopulations. And so within conservation genetics, a very important action or very important question is asking about rates of gene flow between populations. So are populations currently connected? Were they historically connected? And how have these connectivity regimes been altered? Notably, how have humans altered these connectivity regimes? So today we're looking at connectivity of roseate terns. Roseate terns are a seabird with a global distribution, and they are an IUCN species of least concern, but that tends to be a bit misleading about their population status because populations are very patchily and sparsely distributed globally. And then individual populations tend to be threatened and endangered across most of the range of roseate terns. Most of these populations are of critical concern within their breeding locations. And historically, most populations have experienced some degree of range contractions where their historical breeding distribution is actually shrinking and populations are becoming concentrated. And then population declines, particularly at the turn of the 20th century. So here today, we're talking specifically about the Atlantic populations or metapopulations of roseate terns. There's actually 4 putative metapopulations, and today we're only going to be looking at 3. So we're looking at the Northwestern Atlantic population, the much smaller Azores population, and then the Caribbean population. And we're not looking at the upper UK, Ireland population, which also has about 12 to 1,500 breeding pairs. So past work has shown that within the Northwestern Atlantic, there is high rates of connectivity among populations. And we know that from genetic work done by Dr. Stish. And we also know that from pretty extensive banding work done by Jeff Spendelow and Ian Nesbitt and a whole host of people over the years. So within the Northwestern Atlantic, there's high connectivity. However, we don't really know much about connectivity among the other 3 breeding populations. So Anna Loshko did do work looking at connectivity between the Northwestern Atlantic and Azores populations and did find low rates of connectivity or high differentiation between those populations. But the Caribbean population has never been genetically analyzed. And the working assumption is that there is no dispersal between these 3 subpopulations, and that's based on band returns and morphology and different behavior. But we think that that would be kind of unique or unusual for a seabird to have such limited dispersal across what is really relatively small distances. So seabirds are obviously very highly mobile birds, and roseate terns are certainly among them. They're also migratory. So the Northwestern Atlantic population shares a wintering ground off the coast of eastern South America, centered around Brazil. And we know that the Northwestern Atlantic population actually hops down and uses the Caribbean as a staging ground on the way to that wintering location. And then we believe that the Caribbean and Northwestern Atlantic populations then overlap on those wintering grounds, although we have a pretty poor understanding of wintering ecology. In other seabirds, all this suggests that population would be interbreeding because mixing on the wintering grounds is really facilitates dispersal among populations. And then we can see that the Azores population is also capable of traveling long distance locations down to that shared wintering ground on the western coast of Africa. So all this to say that any evidence for differentiation among these 3 populations would, we think, be somewhat unusual and might suggest that there's been some sort of anthropogenic mediation of potentially historical connectivity regimes. So we had some research questions. Today we're just looking at current connectivity as opposed to historical. So we're asking if roseate terns in the Atlantic Basin are currently connected by dispersal. And we also want to look at genetic diversity. So how have range contractions and population declines potentially influence diversity within these 3 sampling or 3 populations. So we actually have 4 sampling sites because we were also interested about dispersal within the Caribbean. There's very low banding effort in the Caribbean, and so we don't have a good idea of dispersal of this large meta population. So we have samples collected by Carolyn Mostello from Bird Island, Massachusetts. We have samples collected by Veronica Neves from the Azores, although only 4 of those samples successfully amplified. And then we have samples collected in the US Virgin Islands by myself, and samples from Florida collected by myself and Ricardo Zambrano. So kind of an overview of these 3 potential populations for sampling regions. The basic foundation of population genomics is that we're using large numbers of these variant sites called single nucleotide polymorphisms, or SNPs, to assess population genetic questions. And kind of the idea is that the more SNPs we have, the more statistical power we have to resolve these population genetic questions and questions of relatedness. But the genome is huge. Our genomes are huge, birds have huge genomes, and we're not really interested in sequencing that entire genome. So the issue is how to restrict our sequencing while still recovering the exact same SNPs between individuals, because we need to make sure that we're looking at exactly the same area of the genome for each variant site to make sure that we're accurately looking at population dynamics between populations. And so there's a lot of ways of restricting sequencing while also accessing or recovering the same variant sites. And we've opted to use targeted sequencing with ultra-conserved elements. And these are essentially these stretches in the genome that are highly conserved. So we can use a synthetic probe to go into the genome, recover this highly conserved site, and return it for us. And then hopefully piggybacking along with this conserved site will be a variant site that we can use to answer population genetic questions. Now it's a bit technical, but we literally are going fishing within the genome. So if we have 3 roseate tern chicks with a fragmented genome, and we're really only interested in looking at this red site that might have a variant site that might be used to help resolve population genetic questions. We actually use this synthetically crafted probe as bait for fishing. So we receive these probes from a company, we send them into the genome, and then we're recovering only that site of interest and not the other sites that we're not interested in. Then we can amplify only the site of interest and get very high read depth, which ensures that we're looking at correct variant sites and not looking at sequencing error. So depth is an important component in the sequencing. Obviously, one little red site isn't going to tell us much about population, so we actually did this 5,000 times. There's 5,000 recognized UCE sites within tetrapods, so we share the same UCEs with roseate terns, with lizards, with everybody. So just briefly looking at our genomics workflow, I won't go into this. I know this might not be of interest to a lot of people, but ultimately we collected our samples and extracted DNA. And then to each individual, as part of the genomic library preparation, we affixed an individual identity. So then once we pulled our samples and enriched them with those 5,000 targets and sequenced them, we could then split them back up. So we're still only looking at individuals, even though we've pooled our sites together. And hopefully we've collected shared sites among those individuals that we can investigate. So overall, we recovered 1,568 UCE loci that were common among all 71 of our individual samples. And that's a pretty common recovery rate for UCE analysis. And that translated to 2,841 SNPs, which is very adequate for evaluating questions of population dynamics, with a mean site depth of 56x per individual. So that suggests we have very high read depth and that we're not evaluating errors made during the sequencing process, that these are true variant sites. We'll start just by quickly looking at genetic diversity. So we'll just look at two metrics of genetic diversity: allelic richness, which is the average number of alleles for all sites within a population, and observed heterozygosity. So within a population, what percentage of individuals have two different alleles at a site versus one allele on both chromosomes, which is representative of kind of low genetic diversity. And we'll start with the Azores. At the Azores, we saw significantly lower allelic richness and observed heterozygosity in the other three sites. We do have a smaller sample size for the Azores. However, these metrics are pretty robust to sample size. So this does suggest that the smaller population does have lower genetic diversity. And then we saw that actually the USVI, which is kind of our largest population, had significantly higher allelic richness than the other sites. So what we might be seeing here, although it's hard to make inferences, is that our larger site had higher genetic diversity and our smallest population had the lowest genetic diversity. Then we looked at FST, which is a measure of pairwise differentiation between populations on a scale of 0 being no differentiation, 1 being total differentiation. But with birds, we might think that an FST of 0.05 is relatively high. Looking first at the Azores as related to the other populations, we see significantly high FST, indicative of genetic structuring. So this indicates that there is barriers to dispersal from the Azores to the other sampling locations. Looking then at Massachusetts, we see lower FST, but it's still significant. So significantly different from 0, again, suggesting that there is some barrier to dispersal from Massachusetts to the Caribbean. And then looking just at Florida and the Virgin Islands, we did not have a significant FST, which suggests that Florida and the Virgin Islands are exchanging migrants, that there's not a lot of population structuring between them. Then we looked at Bayesian estimates of migration, and you can think of these totals as representing the proportion of migrants within each population. So looking at Massachusetts, 96% of individuals in Massachusetts originated from the Northwestern colony, so not receiving a lot of migrants. They're also not sending a lot of migrants out, so very low rate of migration from Massachusetts to those other populations. The Azores had slightly lower self-identification, but is still not sending migrants out to those other three populations, so low rates of gene flow from the Azores to the other city sampling locations. The Virgin Islands has a high rate of self-identification, so 97% of individuals originated from whatever this breeding population is. However, the Virgin Islands is sending out migrants. So we see that 25% of individuals in Florida originated from the Virgin Islands. And there also might be some movement from the Virgin Islands to the Azores, although pretty big confidence interval, so it's a little hard to tell. And then Florida is not sending out migrants, instead receiving them from the Virgin Islands. So this is a look at a structure plot. You've seen a few of these. So this is just a way of looking at genetic clustering and admixture among populations. We can see that Massachusetts is clustering strongly out from the other populations. So structure has told us that there's 2 genetic clusters. However, we do see that the Azores has a high proportion of admixture from Massachusetts, and that might just be saying that our low sample size isn't quite able to resolve that relationship. This is a discriminant analysis of principal components plot. So this is similar to structure, but instead we're using the kind of classic idea of principal components and then adding a discriminant function that allows us to maximize the variation between population. And we're seeing similar patterns. So DAPC has told us that there's 2 genetic clusters and that they're splitting out as Massachusetts being differentiated from the other 3 clusters. Although you can see that Azores is a little more intermediary, so perhaps a little more relatedness to Massachusetts. So very similar to what structures told us. And lastly, we use this DAPC to conduct an a priori population assignment. So on the y-axis, you're seeing individual population assignment. On the x-axis, you're seeing the genetic clusters. So we've told DAPC we have 4 genetic clusters. Assign these individuals to clusters based on their genetic identity. What we're seeing is really high rates of assignment. So the blue crosses indicate population of origin. If individuals were not assigning correctly, we would see a lot of red smearing outside of those blue crosses. Instead, we're seeing that only 1 individual is misassigned, and that's this Florida 1861 individual is assigned to the Virgin Islands, but otherwise we had 99% accuracy, which is really promising. So just breaking it briefly down, because I don't want to run over, we saw significant differentiation between the Northwestern Atlantic, the Caribbean, and the Azores population. We see that the Northwestern population in particular is isolated, so there's no gene flow into or out of that population. This suggests there's a low probability of recolonization of these regions if any population is lost. So if we lose the Northwestern population, it's unlikely that there'd be natural recolonization from the Virgin Islands population. We also saw asymmetrical migration from the USVI to Florida. This suggests that Florida is a sink population, taking in individuals, but not producing migrants that are moving out elsewhere into the Caribbean. We had a high accuracy of population assignment, which is really promising for future research on the wintering grounds. We can collect DNA from individuals there and assign them back to their population of origin for areas that don't do banding like the Caribbean. And there's a need for future work, of course. We use neutral loci, so although populations were differentiated, we don't know if those differences were adapted. We don't know if individuals are on different evolutionary trajectories, and that's a really exciting future step. We also need to investigate if this isolation is contemporary or if populations were historically isolated, and that's actually the next step of my research, which I'm very excited about. So I'd like to thank my funding sources and everyone that helped with this project. And I'd be happy to take any questions.
Thank you, Paige. Really interesting work. I think there's one in the chat from Patty already. Wondering if you or others will be able to use primers to target those same SNPs in future work to expand understanding.
Yeah, that would be my hope, because these UCEs are very conserved. We can certainly use the exact same UCEs to hopefully recover those same sites. So for example, if there were samples collected on the wintering grounds, we could use that exact same analysis to try and identify those populations back, which I think would be pretty promising.
Don Lyons is saying, you mentioned one could sample Brazilian wintering grounds and correctly assign individuals to a regional population. Could this assess relative population sizes?
Potentially. Yeah, so realistically, the population here, so if we're looking at the Caribbean, because we have low differentiation between Florida and the USVI, that suggests the Caribbean would be an entire population potentially, or meta-population. And so it'd be very difficult to resolve that question. So yeah, I wouldn't necessarily make that claim, although it's a good idea.
Sean Murphy's asking, would you think a bump in your Azores sample would result in significant differentiation from the other groups?
From the genetic diversity?
Yeah.
I think so. We also conducted these analyses with microsatellite loci, as you saw earlier with Rachel's work, and we had very similar results. And for that analysis, we had more Azores samples. We had 10 samples. They just didn't work well for the genomic research for whatever reason. And we still saw the same patterns there. So we still saw significant differentiation of the Azores from the other samples. We saw similar patterns to structuring, although microsatellites are less able to resolve those population differences. So while I would have liked a larger Azore sample, and it was heartbreaking to not have it, I think we would still see similar results.
Jeff Splendo is saying, in the last decade, we've had at least 5 different roseate terns from Ireland that would have been seen at the Northwest Atlantic colony, but none have been known
cool.
to nest.
Yeah, and that's certainly, and there's past records as well. And Helen Hayes has reported individuals from the Azores in Brazil. So we know that they do move across the Atlantic, but we don't necessarily know if that's indicative of breeding movement. And this suggests that it probably isn't.
Great. Well, thanks so much for your talk, Paige. We got to move on, but keep an eye on the chat because I see there's some more comments coming in. So, John, I see you on video now.
Yes.
Let me just get the title of your talk. So, John, is it Herbert or?
Yes, Herbert.
Is giving a talk on migratory shorebirds. I'm just trying to get to the title here. Migration strategies and global connectivity of migratory shorebirds. I'm just going to cut it short and let you read the full title. So if you want to share your screen, John.
Okay, good afternoon, everybody. My name is John Herbert. Let me get it. Here we go. Okay. I'm a PhD candidate at Tulane University, and I'll be talking about our research, migration strategies and global connectivity of a neotropical Nearctic migratory shorebird. And this is down on the Gulf Coast of Mexico. Okay, so for migration, we want to know what is happening, especially for long distance migratory birds, because they're occupying different habitats throughout the annual cycle, different habitats. The winter range, they have different habitats during spring and fall migration, and then they're stopping along the way. And then they have different habitat in the breeding period. And we want to know what strategies they're using during migration. So are they energy maximizers? Are they time minimizers? Are they using stopover sites during migration? How are these stopover sites affecting migratory timing? Are they encountering ecological barriers during migration, and how is that affecting migratory success and migratory behavior? And then we want to connect these global distributions, put all these puzzle pieces together. So this is what we're trying to do, attempting to do in our study here. And for our study, we're looking at two pieces of the optimal migration theory from Allerstam 2011. So briefly, this graph here, these three curves represent different food availability at a stopover site. So two rules we're looking at: the global update rule and the expectation rule. So here, number two, the global update rule. Birds will stay longer, birds will stay shorter at high quality sites because they're expecting similar habitat quality throughout their migratory route. And then the expectation rule, up here, number one, they're going to exploit these high quality sites for the resources. They're going to stay longer at the high quality sites because of unreliable or unexpected habitat quality along the route. So these are two rules in the optimal migration theory that we're testing here in this study. So our study, we are going to the northern Gulf of Mexico coast. This is after the Gulf, they're migratory birds during spring migration, northbound migration. They're going to cross this major ecological barrier of the Gulf of Mexico and typically stop along the northern Gulf of Mexico coast. So we are looking at investigating strategies of migratory shorebirds, neotropic, Nearctic migratory shorebirds, specifically the semi-palmated sandpiper. They winter in South America, primarily along the Guyana coast, but throughout the coastal South America. And then they breed in the Arctic, and there's three different breeding populations in Western and Alaska, Central Canada, and then Eastern Canada. And as they're migrating, they take several hops. So they're short distance hops, compared to the longer migratory migrations like a red knot. So they'll make many different stops while migrating through North America. So after a trans-Gulf flight, they're going to stop on the Gulf of Mexico coast. And they like to forage in these muddy wetlands, which can be limited on the Gulf Coast. And so here, this backdrop picture here, this is aerial view of Louisiana, and a lot of the coast looks like this now. It's salt marsh habitat, so that these muddy habitats that these particular shorebirds like, and many other shorebirds prefer, is limited on this coast. So our research questions, we wanted to know what migration strategies are these sandpipers using after crossing the Gulf of Mexico. and stopping on the northern Gulf of Mexico coast. What's affecting them? What environmental variables? What intrinsic demographic variables? Sex, age, body condition, where they're breeding. And we also want to know, how are these wetlands on the Gulf Coast connecting to wetlands throughout North America and the Western Hemisphere throughout their annual cycle. So here we're zooming in on to our study sites on coastal Louisiana at Rockefeller Wildlife Refuge and Grand Isle. These are sites that are directly on the coast, of the Gulf of Mexico. So at Rockefeller Wildlife Refuge, this is a refuge managed by the Louisiana Department of Wildlife and Fisheries. It's a larger habitat compared to Grand Isle, a lot of muddy habitat. These impoundments are managed for waterfowl, but they're drained in the spring, creating these nice mud flats that are used extensively by shorebirds. There's limited public access. And also additionally, it was a low impact from the 2010 Deepwater Horizon oil spill. And then our second site, Grand Isle, it's a barrier island on the Gulf Coast. It is mostly sandy and has some marsh habitat, and it's heavily trafficked by people. It's a public beach, so there's a lot of people using the same habitat as shorebirds. So the shorebirds are being pushed around a lot more than they would at Rockefeller. There's not much muddy habitat. They will have some tidal pools to forage in and the beach to forage in, and even this picture here, I observed them foraging in a parking lot a few times, and so there's limited foraging habitat available there. And additionally, it was severely impacted by the 2010 oil spill. So based on all these factors, we're making the assumption that habitat quality is higher at Rockefeller compared to Grand Isle at these two stopover sites during spring migration. All right, so our methods, we went and captured semi-palmated sandpipers, or I may refer to them as CISA throughout this talk. Through 3 years, in 2017 to 19, we collected variables, determined sex, age, and winter range using known stable isotope signatures from Mizrahi and Peters 2011. We determined breeding population based off of sex and morphometrics, based off of prior research, Grado Trevor et al. 2012. We calculated relative fuel load and also looked at some environmental variables of weather, tailwind, humidity, weather condition, and habitat types of sites. So we're comparing the two different sites of Rockefeller and Grand Isle. And to look at stopover behavior, we use nanotags in the MODIS network to look at duration, stopover behavior on the Gulf, and for connectivity. All right, our statistical methods. We had two different sets of models. One to model length of stay, duration on the Gulf Coast. These models looked at body condition, demographics, and site. And then to look at departure behaviors, we use Cox proportional hazard models, which is again another linear model, which have been used a lot in the medical field, but now it's useful for looking at migratory behavior in animals. And it gets this hazard ratio, which we can see this hazard instead of being in a medical field. In our case, it's looking at the probability of departure. So what is causing these birds to depart the stopover site? And again, we looked at body condition, demographics, weather, and site. So back to our questions, we had some predictions. We predicted they would be using the expectation rule of migration because of known limited and unreliable stopover habitat in mid-continental United States. They would have longer duration at Rockefeller, the higher quality site. We expect some influence of demographics. Males need to establish breeding territories. Adults have more experience with migration, and Western breeders going to Alaska, they have a longer distance to get to fly. So we expected them to have different migratory behavior. And then we also expected to add that to connectivity and from the Gulf Coast throughout the Western Hemisphere. All right, so our results. So first, we found that after the CISA are crossing the Gulf of Mexico, they're arriving on the coast in a depleted state. So their body condition at arrival is low, so mean weight of 22 grams, which is lower comparing to prior studies in the prairie pothole regions in the Dakotas, South Carolina, Delaware Bay during spring migration. Our weights are much lower than these other studies. Definitely suggesting that crossing this major ecological barrier is, it definitely plays a role in the migration of this species. We found that they weighed less at Grand Isle, and our stopover duration for all of our individuals, about 8 days, ranged from 1 to 18 days. So our top performing model for our stopover duration, we found length of stay increased with being at Rockefeller. So almost a 50% longer length of stay at this higher quality site. And then further, we found influence of lower relative fuel load. So a lower relative fuel load at capture. And we didn't recapture these birds, so we're just going off of the fuel load they had at capture. That lower relative fuel load, they had a longer duration, which makes sense. They needed to refuel after being depleted and depleted energy. They needed to refuel in order to continue migration. However, we did find influence at sites still influenced duration with similar fuel loads at capture among sites that were still staying at Rockefeller longer. So it's telling us or suggesting that they are exploiting the resources at Rockefeller. They're refueling as much as they can because of potential unreliable habitat on northbound migration. And then at Grand Isle, they're stopping because they're exhausted. So it's still important. These barrier islands are still important. But as soon as they are ready to move on, they move on at a much quicker pace when stopping at Grand Isle. And then departure. Again, we found our main effects model is the top performing model, but what influenced departure probabilities was stopping at Grand Isle. So at any given day, CISA had were 78% more likely to depart than individuals at Rockefeller. And then some weather influence. Again, they were departing with clear skies and no rain. Interestingly enough, higher relative humidity affected departure behavior. So they were leaving when humidity was higher, which is there's been some studies suggesting that at lower humidity levels that's more energetically taxing for migratory birds because it increases water loss. Interesting, we had a tailwind, which generally in migratory birds, we find that they're leaving with favorable tailwinds. We found slightly the opposite. Here are hazard ratios on this top right graph. This dotted line is the hazard ratio from these models. So above a 1 is they're more likely to depart. Below a 1 is they're more likely to stay. So with tailwind, we generally had calm winds throughout our study period, and then they were departing more or less at null tailwinds. And then at higher relative fuel load, again, that makes sense. As they had more fuel load, they were departing. So around a 0.25 fuel load, that was the threshold when they were more likely to depart the Gulf Coast and continue northbound migration. All right, so our takeaways from this part of the study, that they were following the expectation rule. They were taking advantage of the higher quality habitat. So this high quality habitat on the Gulf Coast after this ecological barrier is really important. And it also indicates that there's potential unreliable habitat, which has been found in other studies for shorebirds in North America. Grand Isle, it's still used, these barrier islands are still used heavily by shorebirds, but maybe it's for more of a quick resting stop because they're exhausted after that trans-Gulf flight. And additionally, we also had relocations from Grand Isle to Rockefeller, and we did not have any birds moving from Rockefeller to Grand Isle, which suggests, again, that there was not enough foraging opportunities at Grand Isle, so they had to move to Rockefeller in order to continue their northbound migration. And we had not much influence on demographics and age, sex, or breeding population. But for a time minimizing bird, it's known that gaining fuel stores is the most important aspect during this northbound migration, and that may be influenced more heavily after crossing a barrier island. The Brown et al. Paper looking at geolocators on semi-palmated sandpipers didn't find any influence of sex, but I will say that we would need a higher sample size for demographics to really tease out the results and seeing how demographics really influence the species during northbound migration. And then quickly, before I get into our connectivity results, a couple of cool findings we found from our MODIS using the MODIS network. This left figure, this is a second-year female. So it's her first northbound migration, and she conducted an incomplete migration. So she stayed on, I tagged her on Grand Isle on 18th of May in 2018, and she spent the summer, the breeding period, on the Louisiana coast, and then headed back south during fall migration. And then on this figure on the right, this is another interesting finding we had. 3 individuals departed Rockefeller on 31 May of last year within 30 minutes of each other. And then 9 days later, they showed up on the Hudson Bay within an hour of each other. So this is, we can't say that they're definitely migrating together the whole time, but maybe they're using the same environmental cues during northbound migration, which is really interesting finding. So for our global connectivity, this figure here, these are our connecting flights that we received using the MODIS network. These dashed lines are northbound migration, solid lines are southbound migration. The average, so for our 18 individuals, so a lower sample size during northbound migration, but they averaged a little over 400 kilometers a day. Slightly faster migration speeds out of Rockefeller than Grand Isle. Again, only 3 individuals from Grand Isle, but that does still suggest that this higher quality habitat may be allowing these birds to migrate faster, which could influence breeding success. Again, more individuals tagged will be needed. And then northbound migration, we found a broader migratory path than what's been previously known. There's not a lot of connectivity data, but from the Brown et al. Paper in 2017, where they used geolocators, they found that the birds going from the Gulf Coast just went right to Alaska, the western breeding population. But we did find that it's more of a wider path, and the birds are going to the eastern breeding population. And then we had our nice southbound migration connectivity flights, which we're collaborating with other researchers, which is another great way to utilize the MODIS network. And then that rolls into our putting all of our connectivity pieces together. That we found that the northern Gulf of Mexico coast, the birds stopping on Louisiana, this is a potential hub for this species during spring migration. So these pie charts here, these are based off of stable isotope data, known signatures from Suriname, French Guiana, and Brazil. And then we had some unknowns, which are most likely from Peru and northern South America. And we're getting birds from throughout the wintering range are stopping on the Gulf Coast. And then using morphometric data and these 3 different breeding populations, the western here in Alaska, central and eastern, that they're dispersing throughout the breeding range. And then we can also connect the wintering range to the breeding population. Suriname is the main wintering range for the species, and they're known to breed throughout the breeding range. And we found that, but we also found more birds from the northern South America and Brazil are more prevalent to breed in the western breeding population. And we did have, we did have more birds from the western to the breeding population. But again, it's this Gulf Coast is potentially supporting the entire global population of semi-palmated sandpipers, which is not really known yet, and more attention, I think, should be placed on the Gulf Coast for this species. So our conclusions, again, higher quality habitat after an ecological barrier allows for more efficient migrations. Barrier islands are still important habitat, but they're potentially used just for quick resting and then moving on because they don't provide those resources, because the species is following the expectational migration. But we need more studies. More sites are needed for the species, putting out more nanotags, getting more of a better idea of what's going on with demographics, expanding the MODIS network. And then here in red, highlighting that this is immediate, these concerns, these conservation concerns are immediate. Hurricanes and sea level rise is a real threat to this coast. This summer, Rockefeller was hit really hard twice by hurricanes. Grand Isle was just hit by a separate hurricane. Those of us that have been lucky enough to visit or work in Southwest Louisiana know what a special place it is for waterbird diversity. So really a concerted conservation effort, collaborative effort needs to take place in this part of the Gulf Coast. Acknowledgements, a lot of great people have helped out our funding agencies. With that, I'll take any questions. Just I'm out of time.
Thank you so much, John. Yeah, we're at 120 or 220. So I would, it looks like there's quite a bit of activity in the chat. If you want to take a look at those questions, and then maybe if we have time after the session, we can open it up for further questions. Okay. So Patricia or Patty Barber on lesser blackback gulls in summer locations of lesser blackback gulls in Pennsylvania.
It's John Murphy giving the talk.
Me, Gail.
Oh, John Murphy's giving the talk. Okay, great.
Let's see.
Or Sean, sorry. It's my hearing.
That's okay. All right. Can we see my slides? My opening?
Looks good. Yep.
Okay.
Thank you.
Let me just move this. All right. So thanks for giving me the opportunity to present some of this work today. This afternoon, I'm presenting results from a study that the Pennsylvania Game Commission took on, and really my co-authors on this presentation are the driving force. Patty Barber and Dan Brawning both deserve highlighted acknowledgement before I dive into the presentation. This study looks to address a fundamental and critical knowledge gap in a species biology, and that's identifying a population's origin of breeding. Basic biological information like this is still needed for many avian species, even in North America, and it's especially intriguing when a species is moving into new areas and expanding its range. And this is such the case for the lesser blackback gull that's found to be wintering in eastern North America. The lesser blackback gull is a widely distributed opportunistic large gull. There are 5 recognized subspecies currently around the world, and this talk focuses on Grailsii subspecies, which historically bred along the Atlantic coast of Europe and wintered in southwestern Europe and northwest Africa. Over the past 100 years or so, it's expanded to breed in the Faroe Islands and extended into Iceland. And then more recently, the bird has become an established breeder in Greenland and concurrently became a common winter resident along the coast of eastern Canada and the United States. So this is really where our story will begin, but you can see on this a Birds of the World map that there's not, Greenland is devoid of any data. So let's take a closer look at some more recently published data about Greenland. Prior to 1990, it was an uncommon visitor. And in a Waterbird special publication in 2016, Bortman and Fredrickson suggested, without a formal survey across the entire island, that it appears that several thousand pairs now nest in Greenland, with a particular concentration in the southwestern coast. Okay, let's see. So over that same time that the Greenland population exploded, lesser blackback gulls expanded their non-breeding range to include large portions of North America, in particular, eastern Canada and the United States. In Pennsylvania in particular, it's been recognized as a central location for wintering lesser blackback gulls. There was a study done to look at the records and accounts from 1996 to 2006, and it looks like about a third of all North American records occur in Pennsylvania. And the lack of information on the breeding origins for Eastern North American lesser black-backed gulls, that's a mouthful, provides a unique opportunity to gain basic knowledge that may improve future management decisions. And it also, for a bird that has continued to spread its breeding and non-breeding range, may be a bit of a precursor to other parts of the globe where this bird, may currently be perceived as a vagrant, and that may change if it follows suit with what we've been seeing here. So we headed to these two locations, Nakamixon State Park and Peace Valley Nature Center, two locations where water rarely freezes over the winter, and it's close to landfills. So it's a great spot for gulls to set up for the winter. And this is where those locations are indicated by that red star. And this is where we set out to kick off a lesser black-backed gull tracking study to try and identify where the birds migrate for the breeding season. So a quick slide about the capture and tagging. Birds were captured in 2018 and 19, mostly in 18. All adults were captured. They were not sexed. That's an important note to put out as we start to look through some of the results. They were equipped with a solar platform transmitter terminal, solar PTT, and they were tracked from their release in March of 2018 and all the way up until October of this year. So this is fairly hot off the press movement data. So a little bit of kind of the initial results of the satellite telemetry. One of the best things that I have to note is if you look in this caption, Mitch Doucette, a birder and photographer, was on an offshore birding cruise off the coast of New Brunswick and took a bunch of pictures of a lesser black-backed gull milling around in late August. Turned out it had a transmitter. He submitted information to the BBL, and the BBL got in touch with us to say that he had photos of our bird out doing gull-like things with its solar PTT on. So most of these photos that you'll see through this presentation are one of our birds with the tag on, out kind of doing its thing in August. So we had 9 tagged birds that provided data for at least one annual cycle, and that amassed to 21 annual cycles that we looked at. Nearly 30,000 high quality locations. And this map shows the distribution of all those locations, and the color coding is just to distinguish between individual. And you note that 7 birds are still transmitting today. And 2 points to make with this kind of smattering of points. All of the birds return to the same general winter area, with the exception of, I think, 2 or 3 occasions where some birds kind of bopped down to the mid-Atlantic and as far down as to North Carolina. But the bulk of the winter points over the 3 years of the study remained in southeastern Pennsylvania and in New Jersey. The second point to make is that you'll see there's some commonly used pathways that track between what we're going to try and decipher as the winter range and the breeding range or summer season of these individuals. So if we take a closer look at that point smattering, we see these 2 general pathways, one moving through the St. Lawrence River Valley and kind of heading back out to the coast, and then one more traditional gull-like bopping along the Atlantic coast pathway. And they both kind of meet up here, right as they near Newfoundland. And then the points kind of go all over the place. And here's Greenland up here with a ton of points, but you also see a lot of points along the Canadian coastline here. Looking at this map, we have locations kind of all over the place. So the next question is, can we assess this location data to suggest that specific locations are related to a bird at a breeding colony? And so we looked at ways to parse this out. of the location data and see what can be constrained into what would be a nesting season. So if we take a look at the first kind of characteristics that we try and evaluate in this data is looking at a summer season, because these birds are headed to locations where there are colonies or birds are not being monitored or surveyed in any way. So we have no one on the other end of the side of the in the breeding territories that could maybe say that, oh yeah, there are birds there. So the first piece of information is looking at this summer season. So a paper that was done for lesser blackback gulls in the UK by Clausen et al. In 2012 did a similar analysis of looking at northern latitudes. So setting a summer season by identifying when the bird settles at a northern latitude. And so if you look at this plot we have here, it's just one individual to illustrate this pattern that we're looking for. And you'll see here it is down in Pennsylvania, and then you have spring migration, and you hit this northern latitude, and there's some fuzzy noise there of it's a seabird moving around. And so we've identified that this segment of the plot to be first summer. Then it's followed by a full migration, and it returned back to Pennsylvania. And then you see it start the second spring migration, kind of does a weird bop up north and then back down and then up again, but then you see it level off again at about the same latitude. So this was a way that we could kind of identify that summer season. And if you look at the birds and their duration of those summer seasons, we had 8 of 9 birds that remained at that summer latitude for more than 100 days. And we used 100 days as kind of a conservative estimate of what a bird might need to arrive at a colony, have courtship, nest building, egg laying, incubation, fledgling. Incubation and fledgling alone is nearly 70 days. So we kind of assigned a 100-day cutoff to really be conservative in trying to put these pieces of evidence together to suggest these birds may be breeding. Oh, and this map on the right here is just an example of what those data look like. So you can see this is a good example, but there's a very central location that the tag locations are going back to. So for some of the results that we saw in Greenland, 9 individuals reached the west coast of Greenland, in particular the southwest coast. And if you remember at the beginning of this, I mentioned that that's kind of the hotbed of activity for lesser blackback gull colonies in Greenland. They spent their summer in areas that overlap with known colonies from that Bortman and Frederickson paper. And so kind of, although these data don't directly identify the tagged birds as breeders, it's somewhat of a safe inference to suggest that these locations are overlapping with known and established colonies in Greenland, that they're likely, and their behavior, as we'll look further along in this talk, is consistent with a bird moving during the nesting season. The problem is, if you look down here in this corner, we have 3 locations that did not make it all the way to Greenland, but that is their northern latitude, and they resided there for many days through the summer season. And so we can't do the same thing with those birds that remained in Canada. We can't kind of overlay them with existing known colonies because there are no known colonies of lesser blackback gulls outside of moving west from Greenland. So we looked into other ways of quantifying area usage during the summer months in order to compare those to some published values for lesser blackback gulls and see if those match up, and could we start to suggest that there may be breeding birds in Canada. So this is just a real quick plot to show this total area usage during their summer season. So we took those summer seasons that I showed you earlier, and we took them for all our birds that kind of met that requirement. They hit a northern latitude and they stayed. And so of those birds, you see on our x-axis are the 3 years, and this is a log value of total area usage, because as you'll see, sometimes when it seems like maybe it's a non-breeder or a failed nesting attempt or whatever it may be, we don't know, these birds like to move around. So you'll see that you have 95% kernel density estimates of upwards of 100,000 square kilometers for some of these birds. But in these hatched bars, these are the Canadian birds. And this cutoff of 6,000 square kilometers is a published value from Thaxter et al. In 2015 that looked at kernel density estimates for birds in the Netherlands, same subspecies in the Netherlands with known breeding. So the breeding status was known, and space use was tied to that breeding status. And they found ranges of 33 square kilometers all the way up to 6,000. So you put that 6,000 cutoff in our data, and you'll see that many of the birds fall below it. Most of the Greenland birds, all but one in 2019, fall below that, that cutoff that we've assigned. And you do have Canadian birds that also fall well below that 6,000 square kilometer cutoff. So let's dig a little bit more into the Canadian gulls. So 2 individual birds out of our cohort reached Baffin Island, and they were centered in different locations, but centered around Frobisher Bay, which is right here. It might be hard to see with this map, but again, there are no known breeding records in Canada. And these birds exhibited site fidelity. So they returned to their locations, albeit the total area usage may have been different, but they returned to those locations in subsequent years. And if we take a closer look at, if we look here, you see this one little teal aqua colored circle, and we're going to dive right in there to really get into the weeds about one bird in particular. So this is a bird on an island called Nuyarn Island. I don't know if I'm pronouncing that right, and I apologize if I'm mispronouncing it. But this is an island just off of Baffin Island. And this is an individual where showing here in this map are 2 years of its kernel densities, 2018 and 2020. And you can see that they're very small total area usage during their summer season. In 2018, the summer season only lasted 64 days, but in 2020, it lasted 105 days. And you can see there, the concentration is here, and it looks like, if you were to guess, there's maybe some foraging locations over here that the birds are headed over to. So this data, when you really drill into this, it's a small sample size, especially right with this bird, right? Because we're looking at one individual out of 2 that were in Canada. But we feel pretty strongly that these data do suggest that there are breeding lesser blackback gulls in Baffin Island. Now we've reached out to contacts, and nobody has much information about birds in Baffin Island. Now, if you look to eBird, you'll see records through the summer. There aren't many, but you'll see records of them bouncing around. But no, there's been no data to clearly suggest that birds are breeding here. But it would make sense because they are following suit with how the birds have been behaving across the globe. So where do Pennsylvania's winter lesser blackback gulls breed? Well, let's just kind of rehash, a tale of the tape. So we have 10 birds, but if you look at these, our final destination, we had 7 birds out of the 10 wind up in Greenland. We had 2 in Baffin Island. An overlap with a known breeding colony. 7 of those birds in Greenland overlapped with known colonies. No birds overlapped in Greenland, but of course that's that's to be expected because there's no known breeding locations. But they did both exhibit site fidelity, both birds in Greenland and birds in Baffin. So that's another kind of check for that they would be there. In Greenland, we had 6 birds wind up spending at least one of their years. They spent more than 100 days at that northern summer latitude, and we had 2 birds do that in Baffin. And then their area usage was at below 6,000 kilometers, square kilometers to infer that they might be tied to a colony and a nest. And 8 of the Greenland birds did, and 2 birds at Baffin both did in different years. So again, I think, we're inferring a lot here, making some assumptions, but it appears that these birds are nesting in Baffin, and what was assumed to be the birds would be nesting in Greenland, and they are. So if we look at the migratory connectivity, just to quickly jump around, this is 1900. This is where this subspecies was recognized as kind of the range, and then they moved over to the Faroe Islands and Iceland and migrating back to that original winter location. Then they get to Greenland, and they migrate back to Iceland. So banding records do show that Greenland birds go back to Iceland. But now we have birds that made a different decision and started to head down further into North America from Greenland to winter, and then of course return. And we see winter birds are continuing to be vagrants along the Atlantic and Gulf Coast. So it really makes you wonder where else these birds might just show up. For example, now they may be breeding in Baffin Island and returning to that same location. So in conclusion, to our knowledge, this is the first study to identify the breeding locations of lesser blackbacks that winter in eastern North America. And our results also suggest that the breeding range of this species continues to expand with evidence of the birds possibly nesting in Baffin Island. Our findings set the stage for more studies to confirm breeding in some of these locations, for example, tracking individuals from breeding colonies in Greenland, tracking wintering birds from other winter areas than Pennsylvania, and also higher resolution tracking data. So, fancier equipment to get better resolute data. I know of colleagues that are planning to do that in Massachusetts, probably this coming March. So should be more to come on lesser blackbacks. And with that, I'd like to thank acknowledged partners who helped out with the data. And again, this photo is one of our birds cruising around the coast of New Brunswick. So, and seems a bit fitting for a Friday the 13th. And with that, I'll take any questions if I have any time.
Thank you so much, Sean and colleagues. That's super interesting. And maybe some tourists that are going up into that neck of the woods could be looking out for those birds in Canada.
Yeah.
So we're at 2:42, but maybe I see there's lots of chats going on here. If people are willing to stick around, how about I read just a couple here? So while entirely believable that lesser blackback gulls are breeding with conspecifics in Canada, what do you think the likelihood they are hybridizing with herring gulls in Canada, like they are in Maine?
Right, that's a good point, and I left that out of the conversation. I should look up who made that in the chat box. But that is yeah. There's a hybrid in Maine that on Appledore that I think for 7 or 8 years nested with a herring gull. So there is a chance that these birds are nesting in locations like Baffin Island with herring gulls. It certainly can't be ruled out with the data we have.
Okay, Dave, do you want to go ahead and take over? Yeah, I would encourage you, Sean, to look at the comments here in the chat box. And I'm going to pass it over to Dave. Thank you.
Great, guys. That's some really amazing talks this session. Thanks. Just going to share my screen. And I just wanted to make a few closing remarks and just some quick acknowledgements. So I just wanted to thank everyone for an amazing meeting, and I'm so happy that we did it. And like, this has just been incredible.